Hello, hello, hello. It's just now 7 p.m. on Wednesday, November the 15th, and I want to welcome you all again to the fourth annual UCSB Natural Reserve System Fall Seminar Series. My name is Andrew Brooks, and I will be your moderator for tonight. Tonight will be the last seminar in our series this fall. I want to thank all of our speakers, including tonight's presenters, for giving us a brief glimpse into all of the exciting research being conducted on the UCSB managed natural reserves within the larger UC Natural Reserve System. If you missed a presentation, the seminars from this year and all previous years are available for viewing on the UCSB NRS YouTube channel. Simply open YouTube, type UCSB NRS into the search box. The UCSB NRS channel should be the first one listed in the search results. Before I kick off tonight's program, I would like to briefly go through a bit of housekeeping. The presentation portion of tonight's webinar will last approximately 45 to 50 minutes, and we've set aside the remaining 10 to 15 minutes at the end of the presentations for any questions that you all might have for either our speaker, Patricia Brown, or our reserve director, Dr. Lyndal Laughrin. If you look at the bottom of your screen, you should see a little button labeled Q&A. You can click on that button and then type your questions into the text window. I'll be monitoring the questions during tonight's presentation, so please feel free to type in your questions at any time. With that sorted, I would like to begin by introducing Dr. Lyndal Laughrin, the director of the UC NRS Santa Cruz Island Reserve. Lyndal will give us all a brief history of the reserve and discuss some of the exciting work that is taking place there. Lyndal will then introduce Dr. Patricia Brown, our featured speaker for the evening. Take it away, Lyndal.
Evening. So the Santa Cruz Island Reserve obviously exists on Santa Cruz Island, but it's one of the few reserves in the university's natural reserve system that the university does not outright own the property. We're on property owned by the Nature Conservancy, and our neighboring partners are Channel Islands National Park on the other 25% of the island, and surrounding us 1 mile out, and also out 6 miles to Channel Islands National Marine Sanctuary. I'd also like to acknowledge the Limo Chumash name for Santa Cruz Island is part of the ancestral homelands of the Chumash people. So Santa Cruz Island is the largest of California's 8 channel islands. It's the most diverse in terms of geological substrates, exposures, topography. And these all translate into a great diversity of habitat types, which results in also a high level of species diversity. I'd like to mention that the field station operations and UC operations started on Santa Cruz Island in the summer of 1964 as the UCSB Summer Geology Field Camp. It occurred again in the summer of '65, and it was so successful that folks approached Dr. Kerry Stanton, the island owner and the Santa Cruz Island Company, to see whether or not we could make this a year-round program and open it up to all disciplines besides geology that might be interested in working on Santa Cruz Island. This was successfully negotiated and started its life as Channel Islands Field Station, operated solely by the CSB campus. At the same time, the Natural Reserve System was being birthed independently within the greater part of UC. Our founding parents were Dr. Ken Norris, Dr. William Mayhew, and Dr. Mildred Mathias. Finally, in 1973, Santa Cruz Island, along with a couple of the other UCSB campus reserves, joined the Natural Reserve System, changed our name to Santa Cruz Island Reserve. So, why are people interested in islands? What's the lure? What's the interest? You can maybe relate this back to maybe the obviously the islands are interesting. They're out there kind of in the mist sometimes. They're kind of exotic, isolation. Probably in terms of the biological world, Darwin kind of opened the door when he visited the Galapagos Islands and then wrote his books on the origin of man and the theories of evolution. So you can look at islands in this sort of a sense of their degree of isolation and how that relates to things that get there, how difficult it is, what happens when they get there, how isolated they are, whether things come and go and they can interbreed, whether they're isolated and become new and distinct entities. The Channel Islands are considered a fringing archipelago in that they're close to a mainland continental source. And if we go back in geological time, back during the ice ages, when all the seawater was held in ice, sea level was much lower. There was actually one big island here off the coast of Santa Barbara called Santa Cruz Island. And probably the distances varied among order of 3 to 5 miles, depending on where you were. Very different from the 20 to 30 miles you would find nowadays, from Anacapa up all the way to San Miguel. We know that they were never directly continual. There's a continuous land bridge across because of the deep channel coming out of the Port Hueneme area, the Hueneme Canyon. It's too deep that no matter how low people drop sea level, you can't expose enough land to make a direct connection across there. So from that sense, all organisms had to get across this gap of water on their own, so to speak. Certainly before humans showed up in this neck of the woods, which now the dates would range around in order of 14 to 15,000 years, and maybe even earlier when we can start considering the dating that's coming from evidence found at sites that are buried in the underwater fringing perimeters around the islands. So how do things get here? Well, they can catch a ride on a log, float out here during a big storm event. Things can swim on their own and make it this far. Things can float in the air, wind dispersed, a lot of plant seeds, a lot of insects, and then flying on their own. So we know that a lot of birds have made it out here. That's pretty obvious. Few that don't make it out here, usually the ones that are fairly sedentary, aren't good long distance flyers, or they don't tend to move very far behaviorally. And when we come to look at the animals, bats come to mind, flying mammals. So that brings us to our talk tonight. We're going to have Dr. Pat Brown enlighten us on what bats exist on the Channel Islands, how they might have gotten here, who's here and who comes in frequently. First met Pat in the 1970s when she just finished her PhD at UCLA and started coming out to the islands to start looking at what bats might be out here. And she's been a regular user of the field station, visitor to this island and several of the Channel Islands. And I've been fortunate to know her as a close personal friend for all these years. So with that, I'd like to turn this over to Pat Brown.
Hello. Let me get my screen up to share. Oh, cannot do it while another participant is sharing. Andy, are you there? I
No, I
see.
think I'm still on here.
Oh, okay. All right. Let me
There we
see
go.
if this works now.
Oop, we're just on
Oh,
there.
it's still on there. Should I try again?
Nope, you're there now.
Okay. Right. Let me start here with Okay, well, I have had a great introduction by Lyndal, so I don't have to spend too much time with that. As Lyndal mentioned, I got my doctorate in 1973 studying pallid bats, and a month after I received my degree, I came out to the Channel Islands and we found a colony of pallid bats, which is what I had studied for my doctorate. But the colony on the mainland had been disturbed. And so this was an ideal place for me to continue my research. Bats are the only mammals capable of powered flight. Uh, they are in the order Chiroptera, and there's over 1,700 species of bats worldwide. They have one pup a year. They can live over 30 years, and the record is 42 at this point with the banded bat. And they do a lot of things for the environment and for people. They eat insects that we consider cultural and pests to us in terms of biting mosquitoes that carry a variety of diseases. And in other parts of the world, they're great pollinators and seed dispersers. So echolocation is one of my main areas of study, and it's where bats send out a pulse of sound, and it may not always be ultrasonic. It sometimes is within our range of hearing and perceive echoes from the sound, and then are able to both navigate, detect prey, even tell what sort of prey it is based on that. So in California, we actually have records of 2 pollinator species, but otherwise, all of the bats in California, all of our 26 species, are insectivorous or eat arthropods. Lyndal mentioned this, and I thought did a very good job of bats arrive by air and sometimes aided by the offshore winds in Southern California. A lot of things contribute to which bats will come over here. Certain bats have better flight than others to make it. But once they make it here, they have to find conditions that are suitable. They need roosts, they need whatever prey, which is different for different species of bats. And you have to have males and females, or at least a pregnant female who has a male pup. And one of the things when we look at bats, we're looking at their ability to disperse. Sometimes we have bats with high aspect ratio wings, sort of the fighter pilots of the bat world. And they can fly. In fact, they're the fastest mammal. They've supplanted the cheetah, and they can fly up to 100 miles per hour. They've been tracked. And they're long distance migrators, and crossing the channel, they do it certainly in less than an hour, maybe even in a half hour. When we look at our Channel Islands, and then the lower aspect ratio wings are the majority of the bats, and some of them have really much broader wings than others and are capable of hovering in flight. So a picture of all of the Channel Islands, and I've been to all but one of them. And you'll see here that Catalina and Santa Cruz, 2 of the larger islands, are the closest to the mainland. And so it's no surprise that they have the greatest diversity of species. These are only 11 of the 18 different species that have been detected on Santa Cruz, and Santa Cruz actually has records of all the bats that occur on any of the other Channel Islands. So survey methods have changed over the last 100 years. They used to use shotguns to harvest bats, but on the Channel Islands, the first bat that's in a museum was captured in 1893. And until at least 1948, there were only 7 species documented from the Channel Islands. And these were presented at the first Channel Islands Symposium. I came out in 1973, started looking in roosts and mist netting, which hadn't been done on the island. And through the mist netting, we added 4 more species to the island. Bioacoustics for me used to mean bringing a 40-pound Raycal high-speed tape recorder and a couple of car batteries to operate it. But that isn't really very portable for taking out to an island. So with the interest in bad acoustics increasing, we got much smaller equipment. So now you can even get an application on your iPhone that'll help you listen to bats. But with echolocation, we've now increased, with the acoustics, we've increased the species list to 18. And that's not 18 bats on Santa Cruz Island all the time. There have been records, and some of these have sort of been present in the 1948 census of bats, but they no longer exist regularly or very sporadically. So on Santa Cruz, the first island on this graph, you'll see that on the far left, S in the gold, that's specimens, either capture and release or in museums. And the green is acoustics, and that's been basically added since the 1990s and more so really in the last decade. So there have been visual sightings on some of the islands without the bat being captured. We don't know what sex it was, but the acoustic records, as you can see, are greatly increasing our knowledge. And the B on there means breeding. So we have a total of 6 bats that have been known to breed, i.e. there were juveniles or females or pregnant females. But there's really only 4 species that appear to regularly breed on any of the islands. These are the 4 smaller Channel Islands, and there's been a lot less work done on Anacapa, San Miguel, and Santa Barbara. The question marks were observations, visual observations that are not substantiated by any collection. On the 3 islands, the question marks and the blue squares meant the California myotis, which is known from all of the other Channel Islands. Small bats were seen flying at night, and it's presumed they were California myotis. The Mexican free-tail bat was sighted but not captured on San Miguel. And one of the more interesting sightings on San Miguel is a red bat, there was a beautiful made with an iPhone of a bat flying which was definitely a red bat. We're not sure which of the 2 species it is, an eastern or a western, and I'll get to why we have western red bats here. And then another interesting collection on Santa Barbara was not actually a whole bat, but a skull of a bat in a owl pellet that Paul Collins put into the Santa Barbara Museum. So when we're looking for roosts, it's easiest to find bats in buildings. I mean, they roost in rock crevices, they roost in trees, they roost in a variety of habitats, but they tend to be attracted to the crevices in buildings. So these are some of the roosts that we've studied on the island, all associated with the Stanton Ranch that was the precursor of the Nature Conservancy in terms of ownership. And some of the buildings date back to the late 1800s, even 1860s. And so the main ranch at the island has the most robust bat populations, and the barn on the lower left, the horse barn, is where we have at any one time 4 species of bats roosting in there. Pallid bats, Mexican free-tail bats, and California myotis. The pallid bats, however, breed, the females and their pups are in an adjacent barn called the cow barn, and that one is where they raise their young. And so I was studying mother-young communication and how I found out from telemetry how the mothers would lead their babies out on their first flight, and the babies would follow the mothers around, learning what kind of prey to eat. And all of the bats, males and females, and the pups, once they could fly, congregated in the middle of the night in the horse barn. And they would form large roosts on the ceiling and sort of digest food. Some of them would come in after foraging and use it as a night roost. So it was an interesting time, and I got a lot of wonderful data from the pallid bats. We also banded the pallid bats, and for several years, I think the greatest recapture was 12 years between banding and recapture. But for a while, I had other jobs and duties and didn't get to come out to the island. So we don't really know the longest, but at least 12 years. And as I mentioned, bats are mammals that have one pup a year. The pallid bat is sort of interesting because it does have usually two pups. The very old and the very young females will only have one pup, but most of them have two pups. And there's a picture of the banded bats in the middle ranch cow barn in the night roost or horse barn. There's also a colony of Townsend's big-eared bats, which is there's really no other colonies currently known on the Southern California coast. It's all been developed, and so this is a truly unique and wonderful opportunity to study them. And they're roosting in a historic adobe at Scorpion Acreage and managed by the National Park Service. Now, you don't just, when you go in a roost, you're not just looking for bats. Guano tells a great story. You can ID a bat based on the guano. You can look at the remains. This is a pallid bat guano. It's quite chunky. And if you ever wonder what's the difference between bat guano and rodent droppings, bat guano is made up of basically sort of crushed insect parts. So you just are inedible insect parts, and you just kind of roll it with your fingers, or if you're not, if you want, and it crumbles right away into all little glistening fragments of insects. Whereas rodent droppings are very fibrous and they roll when you, instead of squash. But in this night roost, not only do you see the guano, but you see remains of the prey items, the inedible parts of the prey items. And these are the elytra of the longhorned beetle there, which is very common and is a preferred prey item. And here's the June beetles that at certain times of the summer are very prevalent, and the bats eat those. There's also, I think we found a scorpion, found it earlier there. Anyways, there's a scorpion claw in there too, and the bats bite off the claws and the tail. And there's interesting research coming out of UC Riverside that they are immune to scorpion venom. And pallid bats and analog to pallid bats in other parts of the world have that same behavior. Now, mist netting really changed a lot in terms of being able to capture bats that don't roost in places we go, like mines and buildings or any sort of man-made structures or caves. But you put a very fine, almost like a hairnet, across roads, across water sources, or any place you expect flyways. The one in the lower left is a building at Prisoners Harbor that has a maternity roost of California myotis in it. And to And we keep thinking there might be Yuma myotis, so we occasionally do set up a very tall mist net, a triple high mist net to try and catch them. On our trips out, we regularly miss the pools. The pallid bats really like to come down to the pool, so that was an important place when I was doing my research on pallid bats out there. And this stream in the lower right, that's the stream that comes from the ranch down to Prisoners Harbor. And that is, when there's water in it like this, it's a wonderful place to mist net because it does form large pools where the bats can come down and dip for water. So you do this at night, and we usually stay up well past midnight when we're mist netting. There's a peak in the first couple of hours after dark in bat activity, and then it tapers off, and the bats go to their night roost and go digest their food, and then come out again maybe in the early morning hours before it gets light. But here's my assistant, Jill Carpenter, who's been in the last phase of this research, which started in 2017. She's been an invaluable assistant. And here she is reaching a Mexican freetail bat gathered in the net, and you can see a pallid bat and a big brown bat, or a little brown bat and a big-eared bat, Townsend's big-eared bat. Now, once you get the bat out, because acoustics will only tell you so much, it'll kind of tell you most of the time what species it is, but there's lots of things that you don't know about the bat, such as if it's what the reproductive condition and sex is, and we have a female in the upper left that's pregnant with 2 fetuses, and then when you catch juvenile bats, you can tell that they're young of the year. They usually start flying at about 5 to 6 weeks of age, and so by the time we go out in July, most of the young are volant. And the way you can tell them from the adults, other than that their teeth are very new and sharp, are the epiphyses in one of the long finger bones. The cartilage doesn't fuse, and that doesn't happen for at least 3 months. So during the summer, we can recognize juveniles that way. Capturing the bats also allows us to take wing punches, which it doesn't seem to bother the bats at all. They don't even flinch. But this is very important in genetic analysis for the bats. We've also been involved in with USGS and other agencies in terms of swabbing for white nose syndrome, which is a terrible disease and hopefully will never make it to the islands. And also look at general health of bats. During COVID, they were looking for COVID virus in bats, which luckily they have not found any in any of our North American bats, that coronavirus. But we've done all kinds of research for ourselves and different agencies. And when you release the bat, then you can record a voucher call for the echolocation signal for that bat. And here are just some of the bats that have occurred on the island. The myotis are the most numerous, the genus Myotis, the mouse-eared bats. And these 2 species are pretty rare, have only been taken on, well, let's see, they've been taken on Catalina and Santa Cruz, but just sporadically. They're not residents. You don't catch them every year. You don't record their echolocation signals. We have recorded them on Santa Cruz Island, but not captured them in the last 5 years of our research there. What really changed was putting up long-term bat detectors. So instead of just the small handheld ones or ones we could set up a night or 2, we now can power the bat detectors with solar panels. And we started doing this out on San Nicolas Island, and we got a very interesting result. We recorded bats that were Western red bats, and recorded bats that were yellow bats, which was a first record for the island. They're known from Southern California now. They roost in palm trees. At the time, there were a few palms out on Phyllis. We're usually looking, when we're looking at signals, we're looking for the lowest frequency and the shape of the signal. The bats can change repetition rate and either enhance their frequency output with harmonics or not. This is a simpler form of looking at the call, zero crossing. There's several different programs that you can use to analyze calls. But this was the very interesting thing. So we had a Western red bat, we had a yellow bat, and then we had this in between in terms of frequency. I called it an orange bat for the time being. When we showed it to Chris Corbin, who's the developer of the Anabat and very knowledgeable about red bats all across the country, because there's eastern and western red bats, he said, we call that an eastern red bat. Now, eastern red bat's not supposed to be in California. So we said, okay, that's very interesting. We'll just have to figure what it was. Right after that, we started setting, in 2017, we set long-term and short-term acoustic detectors out on the island. And you can see one of the small battery operated here, which we put near a netting, an area where we're netting. And that is good with its battery, sometimes for up to a week. Depends how much activity it is and how many insects are also calling and filling up your discs. And so here is this detector is in the Central Valley, very close to the field station. And here's one which is very close to that stream I showed you in the other picture, flowing down to Prisoners Harbor, where a lot of our netting. And after we set up the detectors, we were detecting what we called the Western red bats, but also what we now know is the Eastern red bat. But at the time, it was still, we hadn't gotten the definitive analysis. And that happened in July of 2017 and July of 2018. We caught red bats. I have to say, I caught the first red bat on the island in 1978, and that's the only specimen, and it's in the Santa Barbara Museum. At that time, all bats were red bats. There was no Eastern or Western. That happened in 1988 with the advent of genetics. And they used the Continental Divide as the dividing point for bats were genetically different, east, and they were Eastern red bats, and west of the Continental Divide. And they also, if you took the skull out and looked at it, there were differences in the skull, but very difficult. They are very difficult to tell apart in the hand. So we caught this bat. You can see they have nice furry wings. It was a juvenile. This slide doesn't show it as well as the other one, but there was an epiphyseal gap. And we caught 2 one year and 1 only the next year, 2 juveniles and 1 juvenile, of which we took a wing punch of that one because we were now prepared. And that one turned out by 2 methods of doing DNA analysis, mitochondrial DNA, which really just shows the mother's species. And then recently a full genome was done, and it is truly both its mother and father were Eastern red bats. So now we have an Eastern red bats breeding on about as far west as you can get. So that was a very interesting, very exciting finding. And so we subsequently did record them at long-term detectors that we had helped install on Catalina Island. And this has changed sort of the map. This was an early publication before it had been determined that we actually did have Eastern red bats here. And there were areas of sympatry before, which was there. Sympatry meaning that both Eastern and Western red bats occur in the same place at the same time. And so far now there have been 6 different records from doing genetics, both on museum specimens and rehab bats to rehab and taking wing parts and analyzing it. So there's been at least 6 now. Excuse me. Eastern red bats. So we need a new common name for the species. It appears there's one that subsequently has been identified in southern Arizona genetically, that there may be just along the southern tier, and so that these appear to still be the western red bats in this area. As I said, myotis are one of the larger groups of animals, of bats, and we have 2, the California myotis and the Yuma myotis, which breed. Well, the California myotis probably occurs on all of the islands. The Yuma myotis was only known from Catalina Island, which has large bodies of water, and this bat likes to sort of trawl for insects, and it uses its hind feet and kind of skims the water and will eat mosquito larvae and other emerging aquatic insects. That's its preferred prey. So it's usually only found areas where there's larger bodies of water. So the reservoirs at Catalina were perfect, and actually a Yuma myotis in 1893 was the first specimen ever collected from the islands from Catalina. And all of a sudden, when we started netting here, we'd never caught them before on Santa Cruz. That in 2017, we ended up capturing lactating female Yuma myotis, and now it appears there's a breeding colony, even though there's not very large sources of water except for the chlorinated swimming pool. These are called 50 kilohertz myotis, and the scale is such, but they both end at 50 kilohertz, and it's very difficult to tell the difference between California and Yuma myotis with sound. We also, in July of 2022, caught a lactating long-legged or hairy-winged myotis, which was the first for any of the Channel Islands. And it's a species that isn't that common on the adjacent mainland, and usually is at higher elevations. And it was a lactating female, and while it was in the net, another bat was circling, which could have been her pup. And as I mentioned, these two species were relatively rare. Pallid bats emit a wide variety of calls, including complex communication sounds. Bats don't echolocation sounds to identify one another. They have complex and multiple harmonic, sometimes audible to us, communication sounds. Townsend's big-eared bat is unique because it uses harmonics on a regular basis. That means there's a fundamental and a second harmonic, and it can switch energy. And the way of these sounds, the way they're displayed and analyzed at this point, show the intensity is the lighter portions of the call, and because they're higher frequency, the second harmonics are not picked up as well. Oh, wrong way, sorry. Mexican free-tail bats are ubiquitous when you put a recorder out. They probably commute over the channel on a more or less regular basis. On Santa Cruz Island, I've netted them for many years, captured them. We've always had males. We've never had a female there. On Point Magoo, where I've also done some bat studies, they have a maternity colony on Point Magoo. And as the bat flies, that's not very far. But they've never, this has been a bachelor pad in the barn at Santa Cruz Island. And you can see in their call where you get what's called almost a feeding buzz. They decrease the increase the repetition rate and decrease the duration of the call, and then go back to a search phase call. Hoary bats have this little ping-pongy going up and down in frequency, and they're sometimes audible to people or to people with relatively good higher frequency hearing above 15, 16 kilohertz. And you can see the beautiful fur. These are long distance migratory bats, as Mexican freetail bats and red bats. And the yellow bats, these are bats now that we've never captured on the island, but we have regularly recorded them both on Santa Cruz and San Nicolas. And they're one of the Lazurines, and they also do this sort of ping-ponging with the lower frequency, but at a different frequency. And they're solitary and somewhat migratory. The one very curious call that we've been recording on Santa Cruz Island is a canyon bat. It's the smallest of all of our North American bats. It weighs about the same as a couple of dimes, and it's has a distinctive echolocation call. It usually comes out and flies when it's quite light. If you've been out to the desert and seen bats flying around before it gets dark, it's probably the canyon bat. And it is usually flying pretty low, and it's one of the first bats you capture in mistnet. We've been trying, we haven't caught one yet, but we think they're there, and they roost in rock crevices, and there's plenty of those on the island. The western mastic bat is the largest relative of the freetail bats, the molossids that occur in North America. And it has a call that's audible to most people. It gets down to 10 kilohertz. It sounds like a brown towhee as it flies across the sky at night. And that is here pictured next to the smallest bat in North America, the canyon bat. You can see the difference in size. Mastiff bats, I've been on the east end of Santa Cruz and heard them come across the channel from the mainland, and they could commute that very fast. They could be there in a half hour from the mainland, and there is a roost in the Santa Monica Mountains. And then a very rare bat is the next one down from UMOPS, or the mast bats, and that's a big freetail bat. And we've recorded this on both Catalina and Santa Cruz. And but again, it's a bat that we rarely see in California. So it's one of those long, fast flying bats. So in conclusion, either by one method or another, or all of the three, we now have identified 18 species of bats that occur on the Channel Islands at some time. They are not there all at the same time, probably. And some breed, six breed. We don't know how often that red bats have their babies on the island. We haven't caught them the last few years. And the Volans was our first, and we were hoping that there may be a colony. But that's a large percentage of the 26 bats that occur in California as a whole. So bats now pose 85% of the native land mammal species on Santa Cruz Island. And we recognize their echolocation signals as vouchers if they emit very distinctive calls that are separable from other species. And as this type of recordings increase, we could expect that so will potentially the number of bats, but with long-term recordings, we should be able to sort out resident versus vagrant or just transient species. Analyzing the sounds is a big deal, and I'm very indebted to my partner, Bill Rainey, who is doing most of the analysis of these signals. And this is just the people who have assisted me on Santa Cruz Island. I've had other assistants on other islands, and I just couldn't have done it without everyone's help. It's a lot of lugging of equipment and people staying up most of the night, but we've gotten some wonderful results. And with that, I'll take any questions. Let's see, I need to stop sharing my screen, I guess. Do I need to stop sharing my screen, Andy?
Sure, or I can do it for you.
Yeah, I did. Okay.
So Lyndal, do you want to lead off with the questions?
Well, I think most of these questions can be turned over to Pat. So Pat, What species was it that flies at 100 miles an hour?
That's Mexican free-tailed bats, and they've been clocked in Texas, where they've been studied quite a bit. And they have been an airplane has to fly 100 miles an hour, a small plane, to keep up with them. These are bats that have transmitters on.
Okay, and then Dennis Beebe asked that question, and he has another question. Do you recommend and support the deployment of bat boxes?
Not
Bat houses?
usually. The only time bat boxes are really successful is when you've really destroyed a lot of the natural habitat. And bats like to roost in buildings because you take the trees down or you take the other habitat away. And then they'll often roost in people's houses, and people don't always like that or other structures. So putting up a bat box, especially if you're going to exclude a bat or bats from a building, is really a good way to do that. But a freestanding bat house rarely is occupied, and you have to design the houses for the species you're targeting very often. And some species will never go into a bat house.
Okay, Susan King asks, can you talk a little bit about bats and rabies?
Oh, yes. That's one of the viruses that bats get. And it's the one that's transmissible to humans. Probably when you do go out and catch a lot of bats and test them for rabies from a normal population, about 1/10 of 1% of the bats turn out rabid. And there's some bias to some species, such as Mexican free-tailed bats. The percentage is a little higher, but bats are usually not putting themselves in situations where they come in contact with people. So when you find a bat on the ground during the day, certainly don't handle it without gloves. If you ever are bitten by a bat, try to keep that bat so it can be tested by public health for rabies. But the vaccines, which you can have post-exposure now, 100% guarantee that you're not going to get rabies. So to put it in perspective, foxes and raccoons are much larger reservoirs of rabies than bats are.
Okay, Bobby Offen is asking, can you find differences in a single species of bat on different islands? Or do they differ from the mainland?
So far, there hasn't been any genetic work done. Morphologically, the bats look the same as their counterparts on the adjacent mainland. So there's no great morphological differences. Some of the bats, which once they get out there are more sedentary, such as pallid bats, they are not good long distance dispersers. Very well, if any of the bats are going to speciate, I would say it would be pallid bats. And we haven't done that kind of genetic analysis on them to find out.
Okay, Bobby has another question about maternity colonies. At what time do the female bats segregate themselves into these maternity colonies? What's the reason they're removing themselves from the males at this time?
I don't know if it's that or the males don't want to be around them, but on Catalina we have tracked bats that we capture males and females in the night roost and track them. The males, for the most part, roost singly, and they roost in rock crevices, some as far away as Prisoners Harbor. So they're traveling 5 miles or more to day roost. Excuse me, they day roost in the rock crevices. And then they'll come nonstop to the barn to be in the night roost where they get together and get to chat with the females at all. But in a lot of the bat species, there is just male roosts. And that's, they get together in the fall to breed. And the females store the sperm over the winter and then ovulate in very early spring. And the gestation's about 3 months. And the females then, for warmth and companionship for, I think, just cluster in areas, and they need really warm areas to raise the babies. Whereas the females can't go torpid during the day. They're lactating and they have to maintain their strength for hunting. And so it's a very difficult time for females. And so if you pick a warm roost, you don't have to expend energy thermoregulating. And the warmer it is, the faster the young develop.
Okay. Greg Sater is asking, is hantavirus an issue for bats?
No, no. And it's not an issue for all rodents. It's primarily in deer mice. So bat droppings, the only problem you can have with bat droppings and bird droppings is histoplasmosis, which is a fungus that grows on them. And it's, prevalent in the Central Valley, and some areas don't have it at all. So in some areas you can get histo from going into bat caves and not wearing a respiratory, especially if it's very dusty. Guano itself doesn't transmit it, but it's only when you stir up dust and inhale it.
Another kind of related question from Julie Parker. Do bat researchers need rabies vaccinations to study in the field?
No one can require you to get a vaccine, as we well know. But the most people who work with wild mammals, period, because any mammal, not even wild, domesticated, it's a disease that isn't unique to bats. It's just more spectacular to think of a bat carrying it, but it's not. So veterinarians, people who work with mammals, and who handle them, regularly get the vaccine. I started getting it in 1968 when I started working on bats, and it was a lot more painful then than it is now.
Taylor Vians asks, what's the largest roost size you've observed in any of the islands?
Oh, let's see. I think actually it is the population. I mean, I've done so much more work on Santa Cruz, and I know where the roosts are. And so we don't get large colonies there. So 100 or so animals in the roost. I mean, in the barn at night, when we always count the bats coming out at night using night vision equipment and finger tallies to count, and sometimes using lots of volunteers who happen to be there at the time. And the largest number we've had come out of the barn, the main horse barn, has been about 200 bats, and that's mixed from some pallid bats, California myotis, human myotis, and Mexican free-tailed bats. And they're not roosting in a cluster together. They all segregate in that very large barn, pick what temperatures they like the best. I mean, a lot of free-tailed bats will all roost together. So I think that's the largest colony. We don't have any mines or caves where they congregate, so it's mainly the buildings. And for the most part, relative to mainland populations, they're very small. But the roost for pallid bats on the island is a rather large one, even in mainland standards.
Oh, besides the size, Dennis Beebe is asking, can you summarize the population trends on the islands? Have you noticed any pattern over the years?
I don't think my study was ever designed to look at trends. Initially, it was. And so for the first decade or so, we'd go there on a more or less regular basis and count the bats coming out of the barn. And it was pretty stable. I mean, basically, the amount of foraging habitat defines how large a roost is going to be. So it's the carrying capacity of the environment for any particular species. So the one thing that may happen is that all of the islands have been subject to a lot of grazing pressure from different larger and smaller ungulates, non-native. And as these are being removed or have been removed, what's going to happen is you're going to see a rebound in the natural vegetation. And with that will be a more natural insect assemblage, and that could definitely enhance the bat populations.
Okay. Bobby often asks, she's photographed a bat roosting in Tillandsia at Lotus Land in Santa Barbara. She asks if she encourages large clumps of Tillandsia at her house, would she have a chance of attracting bats to her yard?
Gosh, I mean, I'm not sure what kind of bats were they actually feeding on the Tillandsia. I'd
Yeah.
be very surprised because that's a non-native plant. And we don't have any nectar feeding bats this far north. All of the ones that are common are in San Diego County. I mean, that commonly occur, I wouldn't say they're not even that common, but where they occur, they're along the Mexican border pretty much. So, so I don't, unless there's some insects being attracted to the Tillandsia that are attracting the bats, that would be the link I would see. So I'm
Yeah,
not, I can't answer you.
probably needs a better reason to plant Tillandsia than for a bat habitat.
Yeah.
Okay, Wade Farron is asking about the insect debris pile on the second story of the barn. He's seen that when he was out here over the years. And can you say again what species of bat is most likely responsible for these piles?
Yeah, I mean the pile at one time was very, very large, and they used to use the guano from there. It's all pallid bat guano, and they would use it and put it on the rose bushes in the garden, the fruit trees. I mean, it's a fantastic
Right,
fertilizer.
I can remember picking it up with shovels, putting them in buckets.
And now it's a lot less, but you can use that. I mean, if we weren't shoveling it up or removing it, you can use that as an indicator of how many bats are using it from year to year. That's a very good metric. Um, the the bats leave behind, besides guano, they leave behind the inedible parts of their prey. And for pallid bats, that's arthropods, a lot of the two species of large beetle, which showed the wings, but they don't eat the wings. They don't eat the scorpion tails or pinchers, and they sometimes eat a lot of sphinx moths. They don't eat the sphinx moth wings, and so you can tell kind of, especially if you put fresh tarps over or plastic sheeting over or under the roost, but over the older guano piles, you can get an idea seasonally of what they are eating. They also like Jerusalem crickets. They don't eat the Jerusalem cricket legs or heads. So, and that it doesn't look now like there must be that many Jerusalem crickets because I don't see that in the midden anymore.
Right. When you think of bats, you always think of them flying around in the air and capturing things. But I remember working with you when we put those little glowing little balls attached on their fur. You would see them hopping around on the ground, catching the Jerusalem crickets.
Right. Pallid bats hunt on or near the ground, and they have very large eyes. Their eyes relative to their body size are much larger than ours. So they can see, but they're using actually prey produce sounds to hunt. So it's the sort of scratching of beetles, it's the fluttering of moth wings, and the calling of crickets. And these all cue them into their prey. And so very often they'll stop echolocating altogether when they get close, and then they'll land on the ground, which makes them very vulnerable to predation. And bats that are more likely to be predated in some way tend to have more than one baby. And so that's the twins is the one spare for I think the pallid bats. And the tree roosting bats, like red bats, will have up to 4 babies, but probably only 1 or 2 of those survive because it's a much more precarious life hanging in a tree.
So Barry McPherson's asking what species are the main predators of bats?
Excuse me, what species what?
Are the main predators?
Um, Sometimes owls and shrikes, especially if the bats are roosting in a regular place and they kind of wait them out, and the first ones that come out get predated. Um, but for the most part, predators, natural predators, are not the problem for bats. Domesticated cats take a lot of bats. There are none of those on the island. So bats that come down or close to the ground are more likely to get predated or fall out of a tree, like red bats in windstorms do, and hoary bats and yellow bats. So those species are more fragile in the sense of open to predation, but humans are really the greatest threat to bats and what, our animals and our activities. I mean, I have a rehab bat now that was hit by a car. And well, that was my past one, but I have one now that was mauled by a cat and its wing half eaten away.
Bob Zimmerman wants to know, what effect would climate change have on bat distribution and diversity?
We just had, as Lyndal knows, we just had a conference on the Channel Islands, and that was a whole day topic. And, different species are going to react in different ways. I think bats have a slight advantage because they can relocate more easily than other species. And so if it's too dry, too hot, or too much water, which could occur in some cases, they can move up to a point. At some point there'll be no place to go. They have a fairly wide thermal neutral zone because bats in North America, for the most part, can go torpid. And so they can survive really cold temperatures and find roosts which shelter them from hot temperatures. But if global warming affects, or climate change, period, affects the prey base, that's when the bats are really going to be in trouble. So if you have widespread droughts, or then it's going to affect the food they eat, and that's going to affect the bat populations.
Hey, Dave Jurek wants to know, after all these years for your studies on bats, what would you still like to know?
Wow, that's a big one.
We need another hour.
Yeah, I know. I mean, there's so many things that projects I have to write up, and things that I'd like a lot more data on. I think right now, because not only do I work on the islands, most of my work is actually on desert bats, and the proliferation of solar farms is taking out a lot of bat foraging habitat, and the numbers of bats in the roosts we know are dropping. And there has not been good studies done, both pre and post installation of solar. And we're paving the desert with these panels, and we're putting it on flat lands, and very often productive lands for a lot of animals, a lot of wildlife other than bats. So that's my concern. I believe that we have to get off fossil fuels, but we have to do solar wisely, and taking out natural habitat is probably not the best way. So that's one thing I'd like to do more research on.
We have one final question here. Dennis Evie again wants to know whether you know the species that frequents the Solvang Theater Fest theater during the shows.
I've never been to Solvang, but there could be half a dozen species that might
I think
come
we need
in.
more information, Dennis.
Yes, we do need. Well, we need to put a bat detector there and see what, really, you can go out and buy on Wildlife Acoustics what's called an echometer, and attach it to your Android or your iPhone and record bats and get a fairly decent ID with their software they provide. So that would be a really cool tool to use in the theater in Solvang.
Definitely. On that note, I think I'll hand it back to Andy.
Yeah, that was just incredible, Pat. Thank you very much. I want to thank both Pat and Lyndal for their presentations tonight. I think that was a great way to end the 2023 Fall UCSB NRS Seminar Series. I'll remind everyone again that if you came in late for tonight's presentations, if you've missed any of the previous ones, they're all posted on the UCSB NRS YouTube channel. So go to YouTube, click on that little magnifying glass, type in UCSB NRS, and we should be the first thing that pops up in your search results. So thank you all for coming. I'm sure that we'll do this again in 2024. So sometime in early September, check your emails for a notification about the 2024 seminar series. And until then, I'll wish you all good night. I'll put up a slide with some links if you've got any interest in the UC NRS, the system-wide NRS. There's a web link for that. If you've got any interest in any of the other UCSB managed reserves, there's a link to the UCSB NRS website. And finally, there's that little note about how to find the past presentations on YouTube. So with that, I'll say good night. Thank you again, Lyndal and Pat. Thank you, everybody in the audience, for spending an hour with us. And I hope to see you all again next fall.